Ni hao, or as we would say here in New Zealand, kia ora. My name is Toby Mundel and I work at Massey University in New Zealand. The title of my talk will be Overcoming the Challenges for Women Exercising in a Warming World. Firstly, I'd like to thank the Taiwan Society of Exercise Physiology and Fitness for inviting me to present this lecture. To the Chairman, Vice Chairman, Executive Directors, Council members and all attendees to this wonderful conference, I hope you have a fascinating conference and enjoy the information exchanged. Of course, I wish you all well and to keep healthy and safe in these times. It is a shame that this year's conference had to be changed to an online format, but it is great that it can continue. Make the most of this situation. In terms of what I will be speaking on in this lecture, I will first introduce myself in case you do not know me or my research. Then, I will provide you with the latest research from our laboratory, which is conveniently split into 3 sections. Does ambient heat stress increase the risk for athletic females developing iron deficiency? Are athletic women as reliable as men when it comes to their exercise performance? And do estrogen and progesterone explain the female response to exertional heat stress? I am sure that your English is much better than my Mandarin, but I will try and allow plenty of time at the end of each slide to fully understand and interpret the information. Please, let me now tell you more. I am fortunate enough to have been to your wonderful country twice before. The last time was in 2017 when, as you can see, I brought some of my junior lab members along with me. We experienced your generous hospitality, delicious food, and of course leading research in exercise physiology and fitness. I have made some good friends and excellent colleagues, and I'm happy that some have been able to visit me here in New Zealand. In case you have not met me before, let me tell you a little about how I got here. My undergraduate and postgraduate studies were in the United Kingdom at the University of Birmingham, where I became fascinated with heat stress during my PhD. Following a year of postdoctoral research, I moved to Massey University in New Zealand to begin my academic journey, and where I've stayed since 2007. As you can see from the map, New Zealand's closest neighbour is Australia, but we live quite far away from most other people. However, despite the distance, I quickly saw many similarities with Taiwan in terms of the country and also the people. Here you can see both Massey University, Palmerston North and New Zealand. We have 8 universities, which is not very many, and Massey University is almost at the centre of the country. You can see the arrow pointing to Palmerston North. Volcanic activity has shaped the country, and there are plenty of mountains to see. New Zealand is about 7 times larger than Taiwan in terms of land size or area, but you have about 5 times as many people on your island than we do here. My province, the Manawatu, is known for the Southern Hemisphere's largest wind farm, and our city of Palmerston North has about 90,000 people living here. This includes 1 university, 1 technical college, and a large science park that houses research centres for agricultural, horticultural, and food sciences. If you ever get the chance, please visit us here. You are always welcome to join us for any amount of time. This diagram represents the keywords from my research platforms. As you can see, most of this activity is centered around exercise, heat, and performance. For the past 7 years, one focus has been on the responses of females, and this is some of the information I will be sharing with you. This slide provides the results of climate modeling to predict the climate by the year 2100. On the left are the results of what will happen if the world adopts the strictest environmental policies to reduce emissions and therefore represent the best case scenario that we can expect. In this scenario, 27% of the world's land area and 48% of the world's population will be exposed to heat exceeding a deadly threshold by greater than 20 days every year. Notice that Taiwan can expect between 50 and 100 days above this threshold. On the right are the results of what will happen if the world adopts minimal environmental policies to reduce emissions, and therefore represent the business as usual scenario that we can expect. In this scenario, 34% of the world's land area and 54% of the world's population will be exposed to heat exceeding a deadly threshold by greater than 20 days every year. Notice that Taiwan can expect more than 100 days above this threshold. However, another way of viewing this same data is that as time progresses, a greater number of countries can expect greater days above a deadly threshold, but also that it is no longer the countries close to the equator, in other words, the tropical countries, that experience this deadly heat. Instead, countries further north and south in latitude will begin to feel this deadly heat more regularly. Therefore, this is a problem for most of the world. However, this problem is not only for people living in these countries. Here I have highlighted upcoming sporting events that will most likely feature hot and sometimes humid conditions that challenge the athletes taking part. Of course, we know about the Tokyo Olympics being warned as the most heat-stressful ever, but the World University Games and Asian Games in China, and of course the next Soccer World Cup in Qatar, will also take place under difficult conditions. Two questions are important. Will this heat affect performances? And will this heat affect athlete health? Of course, we should also think about the spectators and all the accompanying teams like coaches, sports scientists, and others. Of course, this talk is about female athletes, so it is important to know how this is changing. The number and proportion of female athletes competing continues to increase. Here we can see how this has changed for the last 3 Olympic Games. Tokyo is forecasted to be the first time that females are at equal numbers to males. However, it is not just sport where women are increasing in numbers. The number of women working physically demanding jobs that incur heat stress is also increasing. For example, mining, forestry and logging, construction, firefighting, and the military. This is fantastic, of course, but then we must ask ourselves, how much do we know about the female's response to heat stress? And this is where we have a problem. As you can see from this graph, I recently searched for all research that was completed in the heat and that featured either men or women. This graph is up to date from 2 days ago. Of course, the great thing is that not only is there more research being conducted looking at this area, but the amount of research looking at women is also increasing. Yet, it is still only about 1/3 of the research that investigates men. So, we have a mismatch between how much exercise is being performed by women in the heat and how much we know about them. Until this is rectified, we are behind. As I mentioned previously, I began this research in 2014 with someone you might recognize, Zhe Huan Lei, or Joe as most people know him. Work from his PhD was published in the Journal of Physiology, and this was followed by my current PhD student Lizzie, who published her first study in the American Journal of Physiology and currently has 2 more papers under review in this journal and the Journal of Applied Physiology. So the results I'm about to show you belong to these 5 studies. I'd like to start by giving you an overview of the design and participants, as this is quite important. I have learnt an important lesson since 2014. Research on women across the menstrual cycle not only takes more than twice as long as on males, but is logistically more difficult. Obviously, I would encourage more research in this area, but also caution any potential researchers to persevere. This data is from 3 separate studies. Whilst one involved women who were taking a monophasic combined oral contraceptive pill, the other 2 involved normally cycling women. However, following hormonal analysis, 7 of these women were deemed to be anovulatory or luteal phase deficient, whilst the remaining 19 were ovulatory. So in total, we had 36 participants. Each cohort completed trials in 2 different ambient conditions, either dry and humid heat stress, or under moderate conditions and humid heat stress. Therefore, we have a range of heat stressful conditions from no stress at all to dry heat where sweating is still efficient, and humid heat where less sweat can evaporate into the environment. It was important to us to not just use one environment. On the right, you can see the absolute humidities or vapor pressure of each environment. The protocol for each study involved a 30-minute self-paced trial of cycling, where our participants completed as much work as they could. This was preceded by either a 12 or 20-minute warm-up at a fixed intensity, which also allowed physiological data to be collected. Here you can see the overall characteristics of our 36 women. They are relatively young, light, lean, have a very good VO2 max and peak power output, and also a good training history. So, iron deficiency is the most common micronutrient disease, with a global prevalence estimated at more than 15%. Compromised iron status is frequently observed in female athletes, especially endurance. Prevalence of iron deficiency for those women exercising regularly is 1 in 3, whilst in the general population, or in men, it is 1 in 7. Like their male counterparts, for these exercising women, iron depletion is a consequence of several exercise-associated factors, including hemolysis, hematuria, gastrointestinal bleeding and sweating, and dietary factors such as suboptimal iron intake likely associated with low energy intake. However, unique to females is the blood loss due to menses, which can, on a monthly basis and accrued over time, lead to iron depletion, iron deficiency, anemia, and of course reduced aerobic capacity. However, over the last decade, research has focused on the exercise-induced response of the liver-derived iron regulatory hormone hepcidin in the post-exercise period. This key regulator of iron status ensures homeostatic regulation of iron in response to systemic iron stores by degrading the iron export protein ferroportin located in the intestine, on macrophages and hepatocytes, thereby reducing dietary iron absorption, recycling from senescent erythrocytes, and sequestering of iron in cells. Exercise acutely increases hepcidin, with a peak observed at 3 hours post-exercise, with the primary mediator of this upregulation being the inflammatory cytokine interleukin-6, and the magnitude of the hepcidin response mediated by the individual's current iron stores. The increase in IL-6 during and immediately following a bout of exercise is part of the acute phase or inflammatory response characterized by a leukocytosis and cytokinemia, alongside the release of other acute phase proteins, so for example C-reactive protein. We have previously demonstrated that ambient heat stress has been shown to exacerbate the IL-6 response to exercise in men. However, it is unknown in women if heat stress exacerbates IL-6 or if this upregulates hepcidin to affect iron transport or storage. This could, of course, be modulated by the menstrual cycle. Therefore, what we did in this first study was our participants visited the laboratory on 4 different occasions, 2 during the early follicular phase and 2 during the mid-luteal phase. So here you can see on days 3, 6, 18 and 21 of the menstrual cycle. Within each phase, one trial was a moderate environment and one trial was heat stress. Just to remind you, these 2 phases are characterized by having blood loss as well as low ovarian hormones in the early follicular phase, or more regular blood volume and higher levels of ovarian hormones in the mid-luteal phase. Here we can see the protocol for this study. Participants came into the laboratory and had a resting blood sample. Next, they completed their 20-minute warm-up, followed by their 30-minute time trial. After exercise, we took another blood sample, and then 3 hours after exercise had finished, their final blood sample. Of course, the ambient conditions were either cool, so 18 degrees, or warm humid, 32 degrees, 75% relative humidity. Probably a combination that you're quite familiar with in Taiwan. The blood samples were analyzed then for several markers including serum iron, ferritin, IL-6, hepcidin, and white blood cell count. Here we can see the results of the power output, so performance. Power output became lower in the heat from 18 minutes onwards as our females slowed down. Therefore, performance was lower in the heat by 5 watts or 3%. This was significant. However, there was no effect of the menstrual phase on performance. Here you can see the core body temperature response before and during exercise. Core body temperature was about 0.3 degrees higher in the luteal phase, and this difference persisted throughout exercise. Exercise in the heat caused core body temperature to be higher by the end of exercise by approximately 0.3 degrees. It would have been much higher, but of course participants reduced their pace in the heat to offset some of this. IL-6 increased following exercise, but this was not dependent on menstrual phase or ambient temperature. Equally, hepcidin increased following exercise, but this was not dependent on menstrual phase or ambient temperature. We also observed that leukocyte and neutrophil counts following exercise, following the moderate condition, was only 30 to 40% the magnitude, so lower, than following humid heat. This was not dependent on menstrual phase either. Regression analysis identified that leukocyte count explained 63% of the variance in the IL-6 response, with platelet count contributing another 9% independently. Serum iron levels before exercise explained 35% of the variance in the hepcidin response, with ferritin after exercise contributing another 28% independently. Therefore, to summarize this first study, ambient heat stress increased leukocyte, neutrophil, and platelet counts post-exercise. IL-6 and hepcidin responses were not increased by the heat. This was probably a consequence of core temperature only being 0.3 degrees higher in the heat due to a decreased workload and performance by our participants. No measures were modulated by menstrual phase, and therefore we can conclude that trained, iron-sufficient females suffer no detriment to their iron regulation in the acute exercise period due to ambient heat stress or menstrual phase. There is still considerable bias against women in basic and preclinical biomedical research, including physiology, as highlighted by the graph showing little, if any, research focusing solely on female participants, which would otherwise be the red color. Anatomic and physiologic characteristics that distinguish the exercise response in women from men indicate a need for recommendations and norms specific to women for exercise testing. Yet, there are still no differences in testing and prescription between women and men, as shown by the discipline's foremost guidelines here. a meta-analysis by Hopkins et al. In 2001 concluded that females appeared to be less reliable than males in exercise tests, which could possibly be attributed to the menstrual cycle. However, this was found to be diminished in trained versus non-athletic women. Estrogen and progesterone play important secondary, so non-reproductive, roles that, according to a specific hormonal mix, influence physiological systems differently in women, such as vascular, thermal, and osmotic regulation. However, women of reproductive age that exercise regularly are more likely to display anovulatory and/or luteal phase deficient cycles, up to 50%, as shown here by a reduced fluctuation in estrogen and no increase in progesterone. Also, the prevalence of oral contraceptive pill use amongst physically active and athletic women is high, at over 50%. Therefore, these two groups actually make up more than half of physically active women. Therefore, when considering research on physically active females, it's important to include rather than exclude these cohorts because of their physiology or endocrinology, in addition to eumenorrheic women, in order to make findings as representative and applicable to athletic females as possible. The purpose of this study was to provide measurement error norms specific to athletic women completing a 30-minute self-paced work trial, whilst the secondary purpose was to determine whether ovulatory or hormonal status or ambient profile were moderating variables. Here you can see our three groups compared, so the ovulatory, anovulatory, and oral contraceptive pill users. Their body weight, composition, and fitness and functional capacity was similar, although the oral contraceptive users were slightly younger and therefore also had a shorter training history. Importantly, the ovulatory group were characterized by higher progesterone concentrations, whilst the oral contraceptive users registered lower estrogen concentrations. The individual results can be seen here as a bland Altman plot. Results presented as homoskedastic, although two clear outliers were identified, circled here in red. Interestingly, the two greatest outliers were two of the best performers. One, a former professional and continental road cycling champion, the other, a national masters road cycling champion. Here you can see the results for intraclass correlation coefficient, Pearson's correlation coefficient, the within subject coefficient of variation, and standard error of measurement. Firstly, the anovulatory group appeared to display worse reliability than the other two groups. However, this is likely a consequence of the much smaller sample size, and when the one outlier was removed, so the one that was circled red on the previous slide, the reliability statistics were no longer any different from the other two groups. Next, we can see that ambient environment appears to have no effect on reliability of the protocol. Finally, the duration of the preload also played no part in how reliable the protocol was. Therefore, overall, reliability was high, clinically valid, and acceptable with a reliable bias of 2 kilojoules or 1 watt. So just to summarize from that second study, aerobically trained women performed 30-minute self-paced trials 2 weeks apart with acceptably low variability. Hormonal or ovulatory status and greater heat and humidity do not moderate this reliability. Therefore, this current study provides a baseline in order to correctly identify the true effect of interventions, for example, training, heat acclimation, hydration, or cooling. A pertinent risk factor associated with heat illness is increased physical activity for sport, exercise, or occupation. When considering exertional heat illness, women are at greater risk than men as they display a higher rate of incidence. Therefore, understanding a woman's response to exertional heat stress remains a priority. Determining the factors that influence the female response to exertional heat stress is not new. Here we have results from the studies with the largest representation of female participants, so a sample size of 36 and 43 women for each study, whereby their thermoregulatory responses to low to moderate fixed intensity cycle ergometry for 30-minute bouts was measured in a range of ambient conditions, from temperate to warm humid and hot dry. Both of these studies used regression analysis to determine which morphological, physiological, functional, and ambient factors explained the variance in the female's core temperature response. They concluded that the strength of the relationships and variance explained was dependent on the heat load. Whilst these important results are valid for a number of women, they are unlikely representative of or applicable to athletic women for several reasons. These studies did not report or account for menstrual phase differences in thermoregulation secondary to fluctuations in the primary ovarian steroids, important as this may differ to less trained counterparts, and we have shown this to contribute to the variance explained for core body temperature. Metabolic heat production in female athletes is likely more than double the values previously examined, and trained women have a greater capacity to deal with such a heat load on account of their enhanced heat loss effectors. Finally, the nature of a fixed intensity protocol is fundamentally ecologically invalid for most sports exercise and physical activity. Therefore, we observed 36 trained women complete a reliable, self-paced 30-minute work trial in both warm-dry and warm-humid conditions, who were distinguished by intra-participant, so between their own early follicular and mid-luteal phases, or inter-participant, so between groups of ovulatory, anovulatory, and oral contraceptive user, differences in their endogenous estrogen and progesterone concentration. We conducted regression analysis, just like the previous two studies, and we hypothesized that the ovarian hormones would contribute significantly towards the variants explained in addition to those identified previously, such as body mass, aerobic fitness, and metabolic heat production. Firstly, we wanted to determine which factors either predict or explain the performance. We used mean power output of the 30-minute work trial. Here you can see the factors that entered the regression. So body surface area, VO2 max, peak power output, sweat rate, and ratings of perceived exertion. If we wanted to predict future performance of a female athlete, then their peak power output is the best predictor, explaining 35% of the variance. However, if we want to explain most of the power output, then both peak power output and their perceived exertion response together are best, accounting for 45% of the variance. Next, we wanted to determine which factors either predict or explain the risk for heat illness. We used the peak core temperature response achieved during the 30-minute work trial. Here you can see the factors that entered the regression. So the ratio between body surface area and body mass, estrogen concentration, the core temperature at the start of the time trial, and power output. If we wanted to predict future heat risk of a female athlete, then their core temperature at the start of the work trial is the best predictor. However, if we want to explain most of the heat risk, then their core temperature at the start of the work trial and their power output are best, but estrogen concentration also explains a small part, accounting for 60% of the variance in peak core temperature response. So just to summarize this final study, peak power output is the best predictor of female endurance performance in the heat. Together, peak power output and ratings of perceived exertion explain 45% of the variance in power output. Core temperature at the start of the trial is the best predictor of exertional heat illness in women. And together, core temperature at the start, power output, and concentration of estrogen explain 60% of the variance in the peak core temperature response. Due to COVID-19, we were unable to complete the heat acclimation study on females we had wanted. However, this will be our next project, where we determine if the menstrual phase exerts any effect and if this causes a detriment to iron levels in women due to the chronic stress incurred. To achieve this, we will perform exercise heat acclimation on at least 5 consecutive days using this protocol, where we aim to increase core body temperature by 1 degree and keep that for 1 hour. Participants will repeat this trial for both the early follicular phase and the mid-luteal phase. So, just to summarize the main results from this talk, and going back to what we'd hoped would be achieved at the start. Ambient heat does not appear to be an increased risk factor for females when it comes to iron deficiency. We've also shown that women are as reliable as men when it comes to measurement error of a time trial. And finally, the ovarian hormones, in particular estrogen, do explain some of the female response to exertional heat. Of course, I do need to thank some people. Here are the agencies that have funded some of this research. On the bottom left, you can see my 2 very important PhD students and my other research team. The top right, I have my collaborator from Japan, Professor Naruhiko Kondo, and of course, to everyone else involved, thank you very much. I thank you for listening to this talk and hope to meet you in person one day.